Hello, today we're going to continue our chapter on bacteria and food poisoning. So remember when we were studying our chapter on yeast, I said to you that often students make a mistake between two key terms, i.e. they make the mistake between requirements for growth and the term growth. So we'll just look at that here for bacteria. So in the instance that we were asked for requirements for growth, always think of the number 5 because microorganisms need 5 key things in order to grow. So we'll just put that here. So no 5. In other words, divide your answer into 5 different parts if you see requirements for growth. So what is actually needed in order for bacteria to grow? So we're just going to list 5. So we've got 3, 4 and 5. So first of all, they will need a food source. Now bacteria, we said already, have absolutely no chlorophyll. So if they've no chlorophyll, they cannot photosynthesize and they cannot make their own food. So they need to get their food from an outside source. They can be parasitic, whereby I'm just going to put key terms here because I will be giving you 2 complete sample answers to learn on bacteria in our weekly grinds class. So they can be parasitic, where they live off living matter, or they can be saprophytic and they live off dead or decaying matter like dead fish, dead meat, raw milk, chicken stock, soups, etc. So they live off dead or decaying matter. Now, I've said to you on a few occasions, ours is a food-based course. So when we talk about bacteria on the home economics course, we're talking about them being saprophytic and living off dead or decaying matter. Secondly, we look at another requirement for growth of bacteria, and that is they need moisture. Now, the thing about bacteria, they cannot utilize ice. They cannot use ice for their metabolism. They must have water in a liquid form. So we just note here moisture. How bacteria need moisture in a liquid form. So in a liquid form for their metabolism. They cannot utilize ice. Thirdly, they need warmth. Now, we've already looked at in our previous lesson the 3 different temperature ranges that bacteria grow on. So we have what are called psychrophiles, P-S-Y-C. The spellings are not going well for me, are they, in this chapter? Psychrophiles, mesophiles, and thermophiles. Now, I'll get it right this time. So we have 5 to 20 degrees centigrade. We have 20 to 45 degrees centigrade. And then we have 75 degrees centigrade and over. Now, these are just general requirements for growth, but I'll be asking you to learn 2 specific bacteria in mega detail, E. coli 0157 and also Salmonella, because on the home economics course the syllabus says we are only required to actually know 3 food poisoning bacteria. So whereas I may have 5, even 6 different types of food poisoning bacteria in your chapter, we're always going to go by the syllabus. So you are never ever going to go into an exam and they're going to specifically ask you to write about X, Y, and Z on Salmonella, or A, B, and C on Clostridium botulinum, or D, E, and F on Listeria. The choice is yours, but you are expected to know 3. We learn the names of 3, and we are going to study 2 in in-depth detail. Now in 2013 on question 3 and 2019 question 3, we were asked to write about 1 food poisoning bacteria that we had studied. Now, it might just have been 1, but I'm covering my tail here, and we need to make sure we always have a backup. So they'd never ask 3 in detail. We certainly need to know 3 examples, but they won't expect 3 in detail, but we certainly need to know 2 in detail. Now, you may remember psychrophiles, we had an example was Listeria. A mesophile, that temperature range between 20 and 45 includes body temperature, 37. So Salmonella goes here. And a thermophile then, we'll be talking, particularly in our preservation section, about a bacteria called Clostridium botulinum. So that's food, moisture, warmth. Next, bacteria need the correct oxygen level. Now be careful here. Sometimes if you ask students, list the 5 requirements needed by all microorganisms: food, warmth, moisture, correct pH, and then they say oxygen. You can't say that because different bacteria have different requirements, and we have 3 different categories here. We have what are called aerobic bacteria. We have what are called anaerobic bacteria, and then we have what are called facultative bacteria. So what do we mean by each one? Aerobic bacteria must have oxygen in order to grow. They have to have it. Secondly, anaerobic can only grow in the absence of oxygen. So there can be absolutely no oxygen in the environment. An example would be tinned food, a completely anaerobic environment, or vacuum packed food. And then we have facultative. Then these particular bacteria can adapt to an environment where oxygen is present or absent. So either or. However, one thing we should know about facultative bacteria is that they even though they can adapt to either situation, they can live with or without. For example, yeast, we said, was facultative. But if yeast had its way, it would much prefer an anaerobic environment. So we have what are called facultative aerobes and facultative anaerobes. Yeast is a facultative anaerobe. Salmonella is a facultative anaerobe. Salmonella is a bacteria that it can live with or without the presence of oxygen, but if it's a facultative anaerobe, if Salmonella had its choice, it would prefer an environment devoid of oxygen. And finally then, our 5th requirement for growth. And a reminder again that these are all very general, and if we're asked to write about them generally. But like I say, we will be learning 2 bacteria in specific detail. And this is correct pH. So what I would do here is I would draw the pH scale again, which we did way back when we started with water. This measures the acidity or alkalinity of a food. So we have 7 is neutral, and then below 7 is an acidic environment, and above 7 happens to be a basic or alkaline environment. So just to give you 2 examples, if we go 1, 2, 3, 4, 5, 6, 7, and then 8, 9, 10, 11, 12, 13, 14. So 1, 2, 3, 4, 5, 6.5. I'm going to take a red there. So 6.5 salmonella. So salmonella likes slightly acidic pH, whereas 7, I'm going to write E. coli 0157 is the strain of E. coli, and it likes a pH of 7. So they are our 5 requirements for growth. So just a reminder, requirements for growth refers to what the bacteria requires in order to grow. We always divide our answer under 5 headings: food, warmth, moisture, correct oxygen level, and correct pH. We have gone through the general requirements, saying that bacteria. On our course, we talk about saprophytes. We looked at in our first lesson on bacteria about their mode of nutrition and how saprophytic bacteria secrete enzymes, for example, into dead fish flesh to break down complex nutrients into simpler substances, which are then absorbed via the cell wall. We know that all bacteria, regardless of type, must have water in a liquid form for their metabolism. They cannot utilize ice. Thirdly, bacteria can be divided into 3 different categories according to the temperature range which they prefer most. This is called their optimum temperature range. We have our psychrophiles, our mesophiles, and our thermophiles. Psychrophiles between 5 and 20 degrees centigrade, mesophiles between 20 and 45 degrees centigrade, and thermophiles like a temperature above 75. Then we have our correct oxygen level, because bacteria are divided here under 3 different categories: aerobic, anaerobic, or facultative. Aerobic bacteria like salmonella, for example, aerobic bacteria like E. coli 0157. E. coli 0157 has to have oxygen in order to grow. Then we have anaerobic. They can only grow in an environment devoid of oxygen. Clostridium botulinum would like to live inside a sealed can of, for example, salmon. And then we have facultative. They can adapt to either situation with or without the presence of oxygen. And we know that salmonella is a facultative anaerobe. Yes, it can adapt to both situations with or without oxygen, but if it had its way, it would prefer an anaerobic environment. And finally then, our 5th requirement for growth is the correct pH. The pH scale is a measurement from 0 to 14 that measures the acidity or alkalinity of a substance. 7 is neutral, neither acidic or alkaline, and E. coli 0157 likes a neutral pH when it's growing. And then we have acidic, anything below 7. We know salmonella slightly acidic at a pH of 6.5. Right, moving on then to our next heading, and that is growth. Now, I said to you when we were studying yeast, if you see the word growth, you always write about reproduction. So I'm going to write that then. So if the term growth is on a question. It could be the growth of yeast, the growth of molds, the growth of bacteria. Describe the method of reproduction. And that is absolutely NB. Now, more than likely, they would write after growth brackets reproduction, but just in case to know that. So we're going to describe the growth or how bacteria reproduce. Now you saw that yeast reproduced asexually by what is called budding. We refer to reproduction in bacteria as binary fission. Now, I just want to draw your attention here to three words, and that is when you're talking about reproduction of bacteria, three keywords that must be on your exam paper is binary fission, number one, asexual reproduction has to be mentioned, which is in our sample answers, and the word mitosis. So I'm just going to put a mark around that. Ideally I want a different colour there so that it stands out. So go for, so just binary fission there, and I'm going to put an NB. So binary fission, asexual reproduction and mitosis must be mentioned when writing about reproduction of any bacteria. So just to be careful with that. So we'll go back over here and look at our reproduction. Now, can I just say to you that again, this is very general, and I'm asking you please to learn the two bacteria I mentioned, E. coli 0157, that is the first of our three. The second one I want you to learn in detail for food poisoning bacteria is Salmonella. And the third bacteria that we're going to learn a bit more about is Clostridium botulinum. So the syllabus says we need to know three bacteria. Our three are E. coli 0157, Salmonella, and Clostridium botulinum. Two of those, Salmonella and E. coli 0157, we will cover in this particular chapter, and then we'll talk more about Clostridium botulinum, our third food poisoning bacteria, when we get on to the section on preservation. So growth of bacteria, i.e. the reproduction of bacteria. So we note that the first thing we have to mention is that in order for growth or reproduction to take place, all suitable requirements for growth, or you can call them conditions for growth, must exist. And they include 5 conditions for growth: food, warmth, moisture, correct pH, and correct oxygen level. And depending on the bacteria you're writing about, you would be specific about each of those. Secondly, The two bacteria we're going to learn about for food poisoning section are rod-shaped bacteria. So Salmonella and E. coli 0157 have this shape. So we're going to say that this is the parent bacterial cell, which has, as I'm looking at it there, a little bit dodgy looking down here at the bottom. And sometimes I find when I start changing a diagram, it gets even worse as I go along. Actually, no, it's improved a little bit. So this is the parent bacterial cell. So I'm going to underline as well, actually, all suitable requirements or conditions for growth must exist. And then the parent bacterial cell divides asexually. Remember, we have three words. So asexually by mitosis. And what happens is the bacterial cell divides in two. Now, be careful here. Initially, when the cell division takes place, actually I'm going to do it small, the two new bacteria end up being half the size of the original parent cell. So two new cells form, and they are half the size of the parent cell. Within 20 minutes, the cells will have reached the size of the parent cell. So there's our parent cell. Cell division occurs, two new cells form, half the size of the parent cell. And then within 20 minutes, this is how quickly this reproduction, which is called binary fission. So we'll just mention that as well, our third word. So where we had budding in yeast, it's binary fission. So binary fission, asexual reproduction by mitosis, where the cells divide in two, divide again, 2, 4, 8, 16, etc. So within 20 minutes, the two new cells, bacterial cells, are the same size as the parent cell, and cell division takes place again. And this time, the two cells divide asexually by mitosis. And this time we will end up with 4 new cells, half the size of the original parent cell. Within 20 minutes, they will all be the same size as the parent cell. And what will happen then is cell division will occur again. So then we will end up with 8 new bacterial cells, half the size of the parent cell. And my diagrams are getting more dodgy as I go along, so just be careful about that. Don't be rushing like I am here. We're going over. So we've 2, 4, 5, 6, 7, 8. And then within 20 minutes, you see what's happening here. These cells are the same size as the parent cell again, and then cell division occurs. Now you can be neater in the exam. and then it divides again. Divides, divides, and so on. And that's our reproduction. So just to recap there, growth of bacteria refers to the reproduction of bacteria. Reproduction of bacteria is called binary fission. You must have that word in your answer. You must also make the statement, in order for any type of reproduction to take place, all suitable conditions for growth must exist. Food, warmth, moisture, correct pH, correct oxygen level. Depending on the bacteria you're writing about, you will then be very specific for each of those 5. We're taking one single bacterium or one single parent cell. Remember, there are trillions in our body, but as each bacteria divides by mitosis and asexually, each cell or bacteria divides in 2, then 2 again, 2 again, and 2 again. However, when the parent cell divides originally, the 2 new cells are half the size of the original parent cell. Within 20 minutes, those 2 new cells are the size of the parent cell, and they divide again. And we end up with 4, half the size of the parent cell. They, within 20 minutes, they're the size of the parent cell, divide again, divide again, divide again. And that will keep repeating once every 20 minutes, as long as suitable conditions for growth exist. Now, before we leave it, we should say one other thing, and that is, during rapid cell division, which is obviously what's happening here, with the bacteria, once every 20 minutes is considered very rapid. We note that toxins are given off. So we'll just go there. So as the bacteria are dividing, byproducts of the reproduction or binary fission are toxins, and the toxins are produced during this rapid cell division. Right, so that's basically our lesson for today, whereby we've looked at the requirements for growth of bacteria, the 5 conditions needed in order for any bacteria to grow. Like I say, we were quite general, and also if we see the word growth, we write about binary fission, which is the method of reproduction, making sure we also refer to the word asexual and mitosis in our answer. So that's the end of today's lesson.